We'll get started now. So good afternoon or good morning, wherever you may be tuning in from. In a few short moments, we're going to be very delighted to hear from our main presenter, Dr. Matthew Might. My name is Troy Evans, co-chair of the UDN Peer. Now, for those unfamiliar, the UDN stands for the Undiagnosed Diseases Network, which is a one-of-a-kind program with clinical sites across the United States. Cutting-edge tools and technologies are used in a collaborative network effort in an attempt to drastically shorten the painfully long diagnostic odyssey. To date, the UDN has evaluated over 1,500 participants and has been able to diagnose a whopping 30%. UDN Peer is the participant engagement and empowerment resource made up largely by fellow UDN participants like myself. We seek to further improve the participant experience both pre and post evaluation, as well as focus our efforts on equally important initiatives such as UDN sustainability. UDN Peer's patient outreach includes things such as a quarterly newsletter and quarterly lecture series like the one we're a part of today. If you'd like more information or to become more involved with UDN Peer, we invite you to connect with us via social media or email us at udnpeer@gmail.com. So without further ado, I'd like to introduce you to my amazing co-chair, Megan Halley, who will be moderating our discussion with Dr. Might.
Thank you, Troy, and thank you everyone for joining us today. My name is Megan Halley, as Troy said, and I am, it's my great honor to share with him the co-chair position for the UDN Peer Group. I am a parent of an adorable 6-year-old boy who is a participant in the Stanford Center of the Undiagnosed Diseases Network, and he remains undiagnosed, but we are grateful that the UDN is there to continue to try to solve medical mysteries like his. I also moonlight as a research scholar at the Center for Biomedical Ethics at Stanford, where my own research focuses on the experiences and needs of patients and families with undiagnosed and rare diseases. And it is my absolute pleasure today to welcome Dr. Matt Might to our Tell Me More lecture series, our second in in this series. I'm just going to tell you a little bit about his quite impressive biography. So Dr. Might has been the director of the Hugh Call Precision Medicine Institute at the University of Alabama at Birmingham, UAB, since 2017. At UAB, Matt is the Hugh Call Endowed Chair of Personalized Medicine, a professor of internal medicine and computer science. From 2016 to 2018, Matt was a strategist in the Executive Office of the President in the White House. And in 2015, Matt joined the faculty of the Department of Biomedical Informatics at the Harvard Medical School, first as visiting professor, and since 2017 as senior lecturer. But many of us may know Matt from his connection to the UDN. Matt's journey from computer science to medicine was inspired by his son, Bertrand. In 2012, Bertrand was the first patient in the world to be diagnosed with NGLI1 deficiency through the UDN. And this inspired Matt to use social media to discover other patients and form a community. And through the community's efforts in science, 2 therapeutics for NGLI1 deficiency have been identified since its discovery, and more are under active development. At present, Matt is an investigator on 2 NIH-supported projects to develop and trial targeted therapeutics for NGLI1 deficiency. And Matt is also the co-founder and chief scientific officer of NGLI1.org. And he was a co-founder and scientific advisor to Paranomics. Q-State Biosciences acquired Paranomics in October 2018, and Matt remains a scientific advisor and board member. So that's just a little bit about the background of this incredible scientist and father who we get to hear from today. I just have to say that it is just such an honor to have you here. I know I personally have been following the story of you and your family for many years, and it's just an absolute delight to hear you talk about your work today. Let me just quickly, before we get started, as a housekeeping matter, when Dr. Mite's going to give a presentation, and I'll turn my video off for that as well so he can have the floor. Please put your questions in the Q&A. We will try to save about 10 minutes or more at the end to field some questions for Dr. Mite. So with that, I will turn the floor over to you.
All right, thank you. Let me share my screen. And gosh, I appreciate the lengthy intro. I forgot I do all that sometimes because mostly I just think of myself as dad. And you know, when I was putting this talk together, I had to sort of decide, you know, what perspective. And since I'm speaking to a lot of, you know, parents and patients, I felt like, you know, I could share more of the parent side of it. So, you know, recently when I've spoken, I focused more on what I've been doing in the context of UAB, and I'll certainly talk about that. But I also will share more of the personal side, you know, my own journey through, you know, and into precision medicine. So originally I was going to do my story first and call this, you know, from parent to principal investigator. But I thought, well, let me just tell you the stuff that's, you know, most immediately relevant for you first, you know, the kind of work that we're doing at UAB, and then I'll get into the what I learned as a parent. I will reiterate a disclaimer right up front, which is that I have no medical training. So if, you know, I really am a computer scientist. Most of my academic career, even now, has been spent as an academic computer scientist. So if there's anything in this talk that looks like medical advice, and there's a lot in this talk that will look like medical advice, it's not, and definitely don't do it. Certainly not without a supervising physician. You know, for most of my career, I was doing things like cybersecurity and high performance computing. I did do a little bit of medical robotics work at one point, but really on the sort of like the safety verification side, no real medicine involved in that. And then a lot really just in fundamental algorithms, data structures, a little bit of AI. And that's really where most of my career was, and then got pushed into it through my son, Bertrand. So what I want to talk about are really three things. The first is, what is this thing, precision medicine, and how does it relate to you as a patient or a parent? What is what I call the algorithm for doing precision medicine, and what does that mean for you? If you wanted to do precision medicine, how would you do it? And I'm going to talk about a step-by-step process that we call the algorithm for precision medicine. And then I want to just illustrate what it looks like in practice. So I'll show you examples of folks that we've worked with. And then you go in, you know, some significant detail on my personal story. So what is precision medicine? You've almost certainly heard the term. It begins back actually 10 years ago in 2011 with the National Academies report. And they defined precision medicine as a small essay in the glossary. But what it really boils down to is this, is that if you're using the specific individual characteristics of a patient to tailor medical treatment to just them, then you're doing precision medicine. And there's an emphasis on getting as molecular as possible in this. So you get very, very high resolution on the nature of the condition to do this treatment. And that's really precision medicine. And you know, it's gone through some iterations since, you know, when President Obama launched the Precision Medicine Initiative back in 2015, you know, there was this explosion of claims on the term trying to say, this is what precision medicine really is. And I spent a lot of my time since that day pulling it all back together into a single unifying definition, which I think of as optimizing health with data. So I think if you're optimizing health with data, you're now doing what we call precision medicine. And I think this includes the original definition of tailoring to the individual patient, because so much of that data comes from the individual patient themselves. So in this journey from, you know, the patient over to the best possible treatment for them, or as Obama called it, you know, the right drug at the right time, you pass through a lot of data and some other stuff, but certainly a lot of data. And the piece of data that got everybody excited, and the reason it's a big part of, you know, the UDN and the rare disease community and the undiagnosed disease community, was that the human genome had become clinically accessible. And, you know, the UDN has, I think, a remarkable rate of converting a genome into a diagnosis. I mean, 30-some% for some of the most challenging cases in medicine show what an extraordinary piece of data this is for coming up with a diagnosis. Now, obviously 30% is 100%, so we have further to go. And really what that means is we need to get you to even more data. And in my career, I found that there's data that's been left on the table in the electronic medical record, all this longitudinal, you know, symptomatic data about patients, that's been valuable. And then all the data outside the patient too, everything that's in textbooks and published papers and databases, all of that needs to be pushed into this optimization process. So I do think of precision medicine, you know, trained as an engineer, as as an optimization problem where we're looking for this peak and we're trying to change things like your treatment and what drugs you're taking and your lifestyle, maybe your diet, your exercise to find the optimal health for you. And, and as we practice it at UAB, this is kind of what precision medicine looks like. We've got treatment, we've got lifestyle, we're looking for optimal health. Now in practice, a lot of what I do is work with rare disease patients. And for them, it looks more like this. Sometimes we get to an answer or a set of answers for possible reasonable treatments, or sometimes, more often than not, in fact, we say, well, we couldn't find an answer right now, but we can make a recommendation on some experiments you might want to conduct, some scientific experiments you might want to conduct. And just to show how this sort of breaks down in practice, I think it's probably 10% of the time, data alone is sufficient to produce a reasonable guess as to what might help. Now, this number is actually up from when we started. It was probably 3% when we started doing this about 2.5 years ago. So 10% is actually a significant improvement. Maybe 90% of the time, we have to go to the bench and do some real science to figure out where to go next. And as a practicing scientist, this is, as leader of the institute at UAB, this is what I asked NIH for. I said, this is the infrastructure I need to help these patients, so please help me pay for it. And in practical terms, you can split precision medicine into two halves. Now, from the UDN, you're more familiar with the precision diagnostic half, where you're trying to go from a patient over to a molecular diagnosis for them. And, I'll mention that briefly because obviously it's a really important one. It's a very important one. But, and there was a period of my life where that was all I cared about. And then it shifted more towards precision therapeutics. So I'll spend most of my time talking about precision therapeutics because I think it's important to know what comes after the UDN. How do you move on towards a therapy once you have an answer? My biggest fear would be that we create an amazing network for diagnosis and then have nothing to do for these patients that have an answer, but no next step. And I think we do have an infrastructure in place now to look for those next steps. So again, the focus is mostly going to be on precision therapeutics and really about the two main approaches to doing it. And roughly these are the rational and the empirical. So rational approaches to precision therapeutics start with that core molecular defect, and they try to back solve then for what will precisely undo the effect of that molecular defect. So this can include things like antisense oligonucleotides, drug repurposing that targets a specific mechanism. There's a lot of stuff you can do. Say, here's the core cause, or gene therapy would be in this bucket. This is how we precisely undo that defect. In the empirical, it's more, let's try stuff out and see what works, but using newfound technologies to create what are called precision disease models. So I'll talk about the empirical first. And in the empirical, again, the goal is to go from this molecular finding over to a treatment. In the empirical, you pass through a lot of things on the way. You pass through experiments and model organisms and work on cells and testing drugs. And hopefully at the other end of this process, you end up with something that you can give to a human being. But I want to take a step back here and just briefly touch base with precision diagnostics again, because the empirical can be quite useful if you don't get an answer from the traditional genomic diagnostic process. So for example, let's suppose instead of getting a diagnosis, you got variants of uncertain significance. So some mutations were found, maybe one or two that seem highly suspicious, but they can't quite close the gap and say, this is really what's causing it. This happens a fair amount actually. And the UDN has infrastructure inside of it for trying to answer this question or trying to close this gap. But it's not unlimited infrastructure. And it involves creating precision disease models. So you can create a fly or a worm, or in some cases be a frog, that checks to see if you take that mutation, which is under investigation, and put it into this organism, do you see something that looks like the human disease? If you do, then you may have resolved that variant of uncertain significance and said, well, now we believe it might be causal. So you up the belief or the evidence you have in favor of it being causal. So the empirical side can actually be a next step if you didn't get the answer you were looking for from a traditional genomic diagnostic process. I would say it's useful if it ended in one or two, not more than that, variants of uncertain significance where there's really strong suspicion. yeah, but not quite the smoking gun yet that one of those two is it. So the UDN does have infrastructure and does frequently build these model organisms to try to do it, but they don't have enough resources from NIH to build it for everybody. So they have to parcel out, and try to rank and guess which cases are most likely to benefit from this approach. And as you'll see in a second, one thing I'm working on is trying to provide additional infrastructure outside the UDN that can help patients that might benefit from a model but still don't have one, whether they would with UDN or not. So in general, the way the empirical approach to therapeutics works is you start with the patient, you extract from them a model. A model in this case, it could be their own cells. It could be a cell line that you've modified with their exact mutation. So it's some other cell line that now mimics them or mimics the disease, or it could be an animal. It could be a fly, a worm, a fish, a mouse. It just really depends on what's in scope. When we do this for the purposes of therapeutics, we try to get really small organisms, really nimble organisms that we can test lots of drugs on, because that's what we want to do. Because our next step is to observe the model and to look for some signature of the disease in that system. Scientists will call this a phenotype. And then we start dropping drugs on it and checking to see if there's something out there that rescues that phenotype and makes the model look better again. And once you've got a system for observing this, you can just throw tons of drugs at it, assuming it's a scalable system. So flies are pretty scalable, worms are pretty scalable, cells are very scalable. Mice, not particularly scalable. You can't test a lot of drugs on mice. If you have very targeted suggestions, you can, but you really need those lower organisms to do this very nimbly. And hopefully something out there rescues. And then hopefully that something also rescues in a human being. And what I want to caution here is that it's not uncommon to find hits when you do these kinds of screens. You may find a few drugs that seem to rescue the model, but you can't jump straight to giving that to a human being. You've got to ask a few questions first. The biggest question is, okay, it works in this model system, but at what concentration? If it's at a concentration that's well in excess of what a human body can handle, it may work to rescue the cells, but it's not going to work to rescue a person, at least not without killing them. And so that's something you have to always ask when you see that it's working in a model system or working in a dish. There's a pretty big gap between that and actually going back to a patient. And there's a whole field of study, in fact, that does. It's called medicinal chemistry. So it's great to get to this point, but by no means are you done once you've found your hits from one of these screens. And this is all part of the broader field of precision disease modeling. And what I'm excited to announce that we actually just got a new center at UAB not that long ago, as part of my institute, called the Center for Precision Animal Modeling, which can do this essentially as a service, for patients and physicians. The model is set up so that physicians reach out and say, I have a patient that needs a model. Can you build one? Can you phenotype it? Can you study it? And then bank it once you've got it, once you've reported the results. So we're building a bank over time of these models, phenotypes for these models. And we don't have the funding from NIH to do drug screens, but we at least have the capacity and infrastructure to do it once we've got the model built. So we've got a trial family right now that's paying for the drug screen on top of the model that's gotten built, so we can check to see if there's any other therapeutics out there that might help. So it's a substantial new award, and I'm hoping this becomes an important part of the broader undiagnosed and rare disease ecosystem, because I know we need this infrastructure. And also, I'll say that there's commercial counterparts to this too. There are companies that are doing roughly the same thing. That'll go from a model all the way over to, or a variant to a model to a drug screen, so that there's a lot of activity in this space. The other things you can do with a model is you can look for other targets, other ways to address the disorder. You can do what's called a genetic screen on it and look for different targets to do to check for drugs against. And of course, you can just test lots of drugs. So that's the empirical approach. The rational approach is very principled. So with the rational approach, the first question you ask is, okay, what is the true mechanism of harm? What is the root molecular mechanism of harm in this disease? And sometimes you can almost read it off from a genetics report. It's not always that simple though, because it's not always obvious exactly what that pathogenic mutation is doing. So for example, you have to make a determination. Is this mutation causing this gene to be overactive? Is it causing it to be underactive? Has it wiped it out entirely, like you frequently see with recessive disorders? Or less commonly, but it certainly still happens, has the mutation in the gene somehow made what comes out of the gene toxic? Has the gene itself become a toxin as a result of the mutation, or really the protein that or product that comes out of the gene has become a toxin of some way? And you have to figure this out because there's very different strategies in every direction. So if it's overactive, you want to inhibit it. If it's underactive, you want to activate it. If it's gone missing, you need to compensate for its absence, or you need to replace it and put it back in somehow. And if it's toxic, you need to eliminate it. And in the drug development world, there's a host of different strategies for doing each of these four things. So that's a big role. That's a big challenge we try to solve right up front when we engage with patients. And we've got a lot of tools for trying to figure out what the impact of a variant is, not just whether it's pathogenic or not, but what is it really doing to this patient? And it's opened up a big role for computation and artificial intelligence. So I'll highlight one of the programs we've built to help us solve this problem, but also really solve the problem of what comes next. How can you make rational predictions of what might be therapeutic? And this program, Medicanron, has been funded by what's called the Translator Consortium out of NCATS. And what it's done is it's given this artificial intelligence agents that we've built, which is, I'll say now, is freely available on GitHub. Anybody can download it and use it, although it's not the friendliest user interface right now. If you have issues, do just contact me directly so we can run the appropriate queries. It's giving us access to hundreds of biomedical data sets to power these queries as we search for potential therapies. I'll tell you right now that the class of diseases we're best suited to go after are ones which are autosomal dominant with a gain or loss of function. Because there we're not trying to replace anything that's missing. We're not trying to get rid of something. We're just trying to adjust the thermostat on a gene, if you will, try to modulate its activity a little bit. So we tend to do best on these, but by no means, should you give up if that's not your disorder. I mean, we have other techniques that are more advanced for trying to find things for other disorders. So what is this tool ultimately? Well, in a nutshell, it's really a fusion of old school logic with high-speed automated reasoning to achieve superhuman-like deduction with clinical insight. And so you can ask it very low-level questions. You can say, hey, what's an inhibitor for X? And it can go off and just search for molecular inhibitors. Or you can ask it very high-level questions like, what's a potential treatment for X? And this tends to work better on more well-studied conditions, obviously. And it can come up with lots of interesting ideas in some cases. So for example, if you ask it about overactive bladder, it comes up with 145 suggestions, which is honestly just a lot, more than I thought there might be out there. But it can tell you why it believes each one might be a potential treatment. So how do we build this tool? Well, we read about 30 million abstracts from PubMed. Of course, we didn't read them. We actually used natural language processing to condense all of them into little bits of knowledge. And then through condensed knowledge bases from these other translator data sets into the mix, started cross-linking them so they could talk to each other. And this created the synthetic brain for Medicanron. And if you look inside the synthetic brain, it's a bunch of nodes, it's a bunch of things, a bunch of nouns, if you will. So if you look inside this brain, you see a bunch of dots, and each dot is labeled with a noun, a concept, if you will, something like a gene, a drug, a disease, a symptom. It's a thing. And then there's edges between these nodes that are relationships between them, like this drug treats this disease. So each node, edge, node pair corresponds to a sentence. It's a fact, a biomedical fact, if you will, that's been pulled from somewhere. So for example, one of the things you'll find in this virtual brain is the drug Prevacid inhibits the target NGase. So that's an example of encoding. And once you've got this stuff in there, you can start to do more interesting kinds of reasoning on top. So for example, regardless of what blue and red are, if you know that blue increases red and red decreases green, you can infer logically that by increasing a decreaser, you're overall decreasing. So blue should decrease green. We can infer that. And so, that's what the brain is inside Medicanron, and just routes through it, looking for logical connections between concepts as guided by an analyst looking for ways to hit a particular target or go after a specific disorder. And then a question for us at UAB is, well, how do we engage patients in this process? Because most patients aren't trained to use these AI tools. And the first thing we've done is we've created a step-by-step process. It's really what we call the algorithm for precision medicine. And the goal of this algorithm is to always report what the next possible steps are on the road to a therapeutic. So it could be diagnostic to get on the road, but there's lots of sub-algorithms, if you will, that can get you closer to therapeutics. And it discusses both the rational and the empirical. And it's not always saying that there's just one next step. In many cases, there's lots of next steps. And so one thing we try to do at the Institute when patients reach out is say, well, if we were to prioritize and try to guess, where the biggest benefit would come from in terms of what you could do, here's how we'd rank your options. We try to be mindful also of the resources that it would take to complete those, because some things are relatively achievable, some things are incredibly expensive, and we recognize that everybody has, in most cases, no or very limited means to do these things. So recognizing that most of us are strapped pretty thin, we are building up ever more purely computational infrastructure, because if it's computational, it's free. It's the cost of electricity at that point, which is nothing. And so the broader algorithm looks something like this. We can go through a diagnostic portion, there's a therapeutic portion, and I'm not going to go into the individual detail here. At the end of the talk, I'll have a link that describes the algorithm in about 90 pages of exhaustive detail, and also includes a glossary really targeted at parents trying to make the jump from the parent to the principal investigator, if you will, trying to understand some of the science behind the condition, or for patients that are interested and able to engage in their own journey as well. So we've used this process now. We've done it with hundreds of patients. We're sort of in the middle. We're certainly in, I think, in the proof of concept stage, but transitioning now more towards a scalability stage. So we don't have enough resources to help everybody that reaches out. So we're getting in an increasingly long queue of people that we need to help. So one way we're addressing that is trying to make the process more efficient so that we can handle more and more patients over time. But it's also clear that there's a really important ingredient in this process, a secret ingredient, if you will, to actually helping all these patients, and that's people. We need people, to serve as the interface to this infrastructure that we're building. And we've really relied heavily on brilliant students to do exactly this. I just, got to give a lot of credit to the team. We have students that really go above and beyond what I think any normal student would ever do, because they serve really, in the middle, helping to coordinate between the patient, their physician, scientific teams, and then the software that we built too, as they navigate towards an answer. So I'll give you some examples now of the kinds of things that we've done and they've done. So for example, one of these analysts actually picked up a patient that had a variant in a gene called TMLHE. She was able to deduce that this was likely a loss of function in this gene, leading to carnitine biosynthesis deficiency, which resulted in a recommendation of supplemental carnitine. Now, the high-level disorder, one of the striking features was seizures. And after routing this back through the physician to the patient and making that recommendation, we heard back that it seemed to result in a noticeable reduction in seizures. So that was that was fantastic. As another example, and this is an actual UDN case, there was a variant of uncertain significance in MAPK8IP3, and we reasoned that there was, at the very least, some haploinsufficiency occurring here. There could be some other things going on as well, but certainly haploinsufficiency. And Medicanon, reported back that retinoids, like vitamin A, can upregulate the working copy of this gene. So that's exciting. And we routed this recommendation back. The physician made the call. I think this is through the Duke site. And we were delighted when about 6 months later, we got an email from the mom who was kind enough to share this video. And it's just, I mean, every time I see it, it just blows me away. Because the thing you need to know about this disorder is that these children have significant difficulties with walking, even standing. And here's a child who's 6 months, I think he's about 5.5 at this point, trying out this high dose vitamin A. Now, to be clear, there's clearly a lot of really good parenting here too, because you've got to put in the hours, the therapy plus the treatment to get to a point like this. But this is pretty astonishing for a child that was to a large degree trapped on the floor not long before this. And then we got another update about a year later. So I guess he's probably about 6.5 from this video. And you know, the scope of the progress between these two videos was just astonishing to us. And so here he is, clearly working hard again and making real progress. So again, it's hard to believe there's just 1 year between these two videos and the progress that he's made. So again, you know, it's certainly our hope, and we're doing our homework, that, you know, the vitamin A helped here. But also you got to give a lot of credit to the parents for putting in the hours to make this act, to unlock all the potential, or actually achieve all the potential that something like vitamin A can unlock in a case like this. So you'll see he even
keep going. You're doing great.
Pick himself back up right after this. So just and like I said, we're doing our homework on this one. This has become a very serious scientific investigation at the Institute to try to determine exactly how this is working, what else.
Oh my gosh, that.
Well, we have our theories as to why it works. We think it upregulates the working copy of the gene. Another UDN case, yeah, that's, you know, been fascinating to work with and has taught us a lot about our own process. In fact, is hyperactive KCNMA1. You may have seen this one on Netflix or the New York Times. What it was a confirmed gain of function in this gene, and resulted in on the order of 150 cataplex episodes per day. So this is just heartbreaking to watch. And about 2 years ago, in an early version of Medicanerin, they said, well, what might inhibit this particular potassium channel? Because it's a potassium channel that's doing this. And it came up with a suggestion in 0.028 seconds. So all you have to do is give this child crystal meth. Things should be good to go, because crystal meth will inhibit this particular channel. All good. Sadly, we laughed this off at the time. I don't even laugh off. We just sort of said, oh, well, we can't give a child crystal meth. That's too bad. We got more excited by some of the more exotic things that it found, like spider venoms and plant toxins that are also inhibitors of this channel, but also venoms and toxins. We thought, well, maybe we can modify these and make them into drugs. In tandem with that, working with a company, we did a chemical screen. While all this was going on, 3 more patients popped up around the world. And when I spoke to the dad for one of them, he goes, oh, by the way, we found a pretty successful treatment. I go, what? Well, what is it? What did you find? And he goes, oh, it's amphetamines. I go, oh, of course it is. It's Listex amphetamine in particular, which is Vyvanse. It's an ADHD drug. You realize it wasn't the meth part of the methamphetamine that made the difference. And I mean, there's plenty of methylated drugs out there. If the meth made the difference, then, you know, this was the wrong, you know, we should have realized it wasn't the methamphetamine. Something about the amphetamine structure itself was probably going to be helpful. So now we have the ability, when we get drugs that are out of bounds, to check to see if there's anything similar to that drug that is in fact in bounds. And you know, the structural similarity here is quite high in between methamphetamine and regular amphetamine. It really is just a methyl group. So now, when I last spoke to the patient community, I hear there's now 5 supposedly treated successfully around the world, and they get a dramatic reduction, almost total cessation of these cataplexic episodes with this. So we've been following up with some of the treating physicians and the team at NIH. We'd like to do a much deeper investigation of these patients to see if we can identify exactly how this is working and to what extent that it is, and if there's other things that might be out there that could work for them. Another key milestone that we've reached is, you know, a recommendation from the AI tool proceeding all the way to an FDA trial. So some of you may know this mom. She's a wonderful active advocate in the rare disease community, head of the ADNP Foundation. She reached out and said, is there anything you think you can do for ADNP? And honestly, at the time, Medicanerin wasn't mature enough to give a meaningful answer for ADNP, but a year later, with a year of progress in building in the tool, it was. And it said, you know, maybe you can do low-dose ketamine to upregulate the wild type ADNP that's functioning. And we passed this back to the chief scientific officer of the foundation. He, you know, initiated in conjunction with the families and the foundation, some really deep dives into the literature and the research. I think they did about a year's work of really solid scientific homework on this. And so this for me is sort of like that dream collaboration where we just have to come up with the idea, and then the patients, the parents, the foundation, they can run with that and take it all the way to a clinical trial. And they did. So, you know, by February 2020, they actually had a clinical trial all set up at Mount Sinai. I think we all know what happened in February 2020, so things got a little bit delayed, but I think that, yeah, by September, they actually had it going. They actually had it launched, and we're starting to treat patients with this for ADNP. So we're excited to see that, to see them actually enrolled. And I don't know the results of that yet. I don't know if there are results, but even getting that far was exciting for us. You know, I'll say that, yeah, we've even gone beyond the genetic in some cases. You know, we've had a case of intractable cyclic vomiting where this 19-year-old woman had her parents reach out because she was suffering from multiple episodes of this vomiting per day. She was down to 78 pounds, and you know, parents in absolute desperation. Now, clearly here, there wasn't an obvious genetic cause for us to go after, but they said they tried all known treatments, and we thought, well, that's probably not true. There's probably a lot of treatments you don't even know about, that we don't even know about, because there's 30 million papers out there, and 64,000 of them are on nausea. 4,000 of them are on treating nausea, and between those, there's 347 distinct ways to treat it that are proposed. So for example, if you break these down, you'll find that for Zofran, there's 169 papers. That is the #1 most studied thing to do for nausea. But if you rank these treatments by how well known they are, by how published they are, you start to trend into a region on this graph that we call the unknown known. The things that are published that are technically known by human beings, but not widely known by human beings, and even by practicing clinicians. And if you go way down this curve to where there's about 3 publications in support, you get to nasally inhaled vapors from isopropyl alcohol. And when she tried this, she actually went 8 months without vomiting, able to arrest every episode of nausea thereafter, and even to this day. But 8 months, something else happened. You know, she was up to 125 pounds, but more than that, she was able to marry her high school sweetheart and move to San Diego. And she's been kind enough to allow us to share her story with others as an example of, you know, what we really hope to achieve long-term for everybody with this program. So now I'll transition to why it's me telling you this story, why I'm, you know, running this institute, doing what I do. And it really comes down to my son, Bertrand. And I started off where a lot of you are, really all of you are, or all of you were at some point, on Undiagnosed Island. And when Bertrand was trapped there and we were trapped with him, we didn't know why he had a movement disorder. We didn't know why he had seizures. We didn't know, you know, why he had extreme developmental delay, and why for some reason he could not make tears. So he could cry, but he would never make liquid tears. His eyes were chronically dry, which led to all sorts of problems. And we were on a 4-year diagnostic odyssey, in fact. In the middle of that, we got some terrifying news. You know, they said, the medical team was saying, well, we've got some really bad lab findings. I think I remember saying at the time, it looks like his heart, his brain, and his liver are in a race to kill him first. But I blogged about it because I maintained a blog for him back then, just to keep people informed. And what I said is, you know, we don't know how long he has, but some searching now from what the physicians are telling us indicates he has about 2 to 3 years total. So, you know, we're holding on to what little hope is left, but it seems the windows for miracles is about to close. Now, of course, we'll do everything possible for him. And if that fails, then we'll try the impossible. And that was just kind of the attitude I had was, you know, it doesn't really matter what the odds are. We just got to try and see what happens. Now, you know, 4 years, you know, 4 years in, at 4 years old, we finally got an answer. And it turns out that through exome sequencing, and this is really sort of a prototype of the UDN. It was at Duke, which eventually became part of the UDN. Yeah, this is one of the first ever, I think it was the first ever clinical trial to use exome sequencing in an undiagnosed cohort. And actually this trial itself became part of the inspiration for the broader UDN. And they found he was the first case ever of NGLI1 deficiency, which was definitely an unexpected finding. They realized he was the only then known patient in the entire world with this brand new genetic disease. And did a lot of work. I'm skipping over an awful lot now. You know, built up a huge network of collaborators, many of whom are actually part of the UDN and have become good friends. And they helped me really understand and push the science on this disease. And eventually, about 1.5 years later, realized that as a consequence of this genetic defect, Bertrand was missing something. There was a missing metabolite that he was going to be deficient in. It's called N-acetylglucosamine. And so I Googled it. Yeah, I didn't know much about organic chemistry at the time. I've learned more since, but I could Google things. And so I did Google it. And then what I found out very quickly was you can buy it on Amazon. You can buy the thing that I was pretty sure he was missing on Amazon. So I bought it, got a whole bag of it, ate the entire bag in one sitting, and I didn't die. And for me, that was effectively FDA phase 1 safety testing. And once I wasn't dead from it, I decided it's time to figure out what this could do for Bertram. Now, there was actually 3 weeks of ethical deliberations about this before we finally did it. But eventually realized that, if this thing had been sitting on my shelf and I'd never tried it and something had happened to Bertram, but there might have been some way it might have improved his life or done something for him, I don't think I could forgive myself as a parent for not doing it. So, and also at that point was reasonably convinced it was safe. So I gave it to Bertrand. About 3 days later, walked into his room, and really for the first time in his life, he was actually crying liquid tears. And so I didn't know what to expect from this experiment. I had no idea what was about to happen. I was hoping for maybe better seizure control or something like that, but what we got was tears. And I did what any parent does when they see their child cry for the first time, which is I collected those tears on dry ice and I shipped them to a lab in California for analysis. And those tears from Bertrand, which you've cried many since, really swelled into an ocean of science for the disorder as a whole. And partnered right away with some biologists, because biologists always say, well, it's great that you can help people, but we're biologists. And what's important to us is, can you do it in flies? And so we did it in flies. We actually gave flies NGLY1 deficiency, and we gave them this compound. And sure enough, they went from dying at high rates, like 80% of them would die, to almost all of them would survive. If you want to know more about how we did this and what we did, we ended up publishing it. So Clement Chow was my major collaborator here. And then I wanted to know, can I do more than make Bertrand cry? And it turns out you can. So I was lucky enough to get a grant to do real drug development for NGLY1 deficiency with some collaborators back at the University of Utah. And through a bunch of computational screening, we ended up finding about 70 compounds that looked like they hit just the right target for Bertrand. And 14 of them were already FDA approved. We took all 14 to the bench and we tested all 14 in models. And one of them actually worked in the model. And that one was Prevacid. So if you want to know more about this work, we published that too. And everything we did on the road to finding Prevacid. So it's very specific, obviously, to NGLY1, but there's a process described in this paper that you can extract from it and that we apply over and over. So what did this do for Bertrand? Well, I think it measurably improved his quality of life. Nothing made him, perfect or normal, whatever you want to call it, but better definitely happened. Happier definitely happened. He had, by the end, no seizures, certainly no clinical seizures anyone could see, nothing on the EEG, and his vision was fine. Nothing wrong with his eyes. But more than that, he was happy all day, every day. I think that's what really matters at the end. I think the key metric in life itself is not one we measure in seizures per day or even years per life. It's actually in smiles per hour. It's how are we in terms of our happiness in the moment. But it did other things for Bertrand too. Started unlocking some of his potential. He started making gains in terms of communication. He finally qualified to use an eye gaze computer. And I think it was about a year and a half ago, I walked in the door and he was able to say, hello, like, that is my dad, which he'd previously not been able to do. But everything was always tenuous with Bertrand. It was never easy. And I'll share, probably one of the darkest moments I had with him, which was back in May of 2019. I'm sure you're all familiar with something like this. I took him and his siblings over to the science museum for the day, looking at the fish. And then 5 hours later, we're in the ER and he's almost dead from septic shock. And it was the launch of this second diagnostic odyssey, a 5-week ICU stay. I mean, it was just awful. And so I just, jump into action saying, okay, what's going on here? I asked Mitty Cameron 3 questions. I said, what causes septic shock? What are the consequences of septic shock? How do we treat septic shock? Because I'm not a doctor. I don't know what septic shock is, but suddenly I did. I said, well, you got to, and one of the results was check albumin. And I knew from his medical history, Bertrand, in all the patients that have NGLY1 deficiency, all have low albumin. And this is a critical predictor for survival in septic shock. So I told the team, here's a paper. This is why he needs it. Here's 2 papers, in fact. This is why he needs albumin right now. And on that very first night, he got albumin and he survived the night and started this long odyssey. Got to meet some wonderful infectious disease docs as we hunted down what was going on. Unfortunately, all the cultures kept coming back negative. But slowly over time, he was starting to stabilize. And by week 2, he was doing, still not in great shape, but doing slightly better than he'd been doing. And he even got out for one day with supplemental oxygen and a minor fever. Unfortunately, 24 hours later, his leg was in extreme pain. He was right back in the ER, and we were on to week 3. And it looked like his bones were now infected. He had a bone infection in his leg. He had ascites, and suddenly also had a pleural effusion and a collapsed lung. Unfortunately, the cultures were still negative. It took 2 surgeries to clean out the bone. He needed a chest tube to clear out the pleural effusion. I remember, like, the day that he was supposed to get the chest tube, he looked like this in the morning, and then 4 hours later, he ended up coding. And I said, well, now he's too sick to actually go in for the procedure to get the chest tube. And so he was in bad shape. And I remember thinking, like, I'm not sure he's even going to survive this night. And I remember sort of basically saying, if this is it, trying to say goodbye to him that very night, because his hematocrit had plunged to 19%. So by the morning, I said, we can't even do the procedure that we want to do without doing another procedure first. He needs to get a blood transfusion now. And somehow he made it all the way through. He got that first chest tube, but they put it in the wrong place. So he coded again. We got another chest tube. And unfortunately, cultures were still negative. And then Memorial Day weekend hit. And I remember the resident, and he was stable, but he's just sort of barely getting along. And the resident said, well, yeah, nothing happens on weekends, and especially nothing happens on holiday weekends in a hospital. And I thought, well, I've got to do something. I've got to do something here. So I wrote code. I added a diagnostic extension to Medicanron, so you could feed it in symptoms, and it would try to guess the disease. So you could give it symptoms, and it could try to find diseases that explained as many symptoms as possible. So if blue are the diseases and red are symptoms, it could rank them and say, here in the order of explanatory power, here are the diseases that it could be. And so here's the initial list that came back from of the top hits for Medicanron. We did aggressive testing for these, and at the same time, dove into something that a lot of you are familiar with, which is genomics, but in this case, metagenomics. And your metagenome is you plus, it's all your DNA plus all the bugs inside of you and their DNA. So you can extract a fluid from a patient, or really any kind of tissue, and you're going to get some mixture of person plus microorganism. And those microorganisms also have genetic material. And so the question is, can you use this stray non-human genetic material to figure out what's infecting somebody? Can we find the bug? And I actually, it took me a day to get it checked out of pathology, but got fluid from Bertrand checked out into boxes that I could take and drive halfway to Huntsville to meet my deputy, Andy Krause, who took it the rest of the way to Huntsville to get it to Hudson Alpha to do metagenomic analysis. They did 40x shotgun sequencing 4 times over in different ways on these samples over the course of 3 days. And then we had a mountain of raw data analyzed to try to pull out the bug that was causing the disease. So it was like the ultra high-speed version of genomic diagnostics. And what I could do is I started producing visualizations of this data. So here is a phylogenetic distribution of all the organisms that were inside Bertrand. And right away, we could see what was not in him from this list. And so we could start crossing things off right off the bat. And it turns out that only one thing was left at the end. Another question was, can we prove that it is what we think it is, which is to say Pseudomonas? And we came up with a technique on the fly to localize the pathogen and prove that this is what it really was. And what it amounted to was a novel way of bursting all the cells in a sample. And I'll just sort of gloss over this now because it's not that relevant. But we came up with a technique on the fly to show that Pseudomonas was present in the fluid in the lungs. And it was present in very, very large quantities. That's what we did with this on the fly experiment. And you're just looking at a little pie chart here showing when we did this enhancement, the enhancement of the Pseudomonas in the sample. So we're pretty sure it was that. We also saw in the viral genetic material, a phage, which is a virus that feeds on bacteria, a virus that only eats Pseudomonas was abundant inside Bertrand's body. And so if you're a virus that only eats Pseudomonas, and there's a lot of you, there must be a lot of Pseudomonas that you're eating. And so reasonably certain that this is what it was. And it also pointed directly to an antibiotic that could work, which is meropenem. And so after 4 weeks in the ICU, within 48 hours, he was out. The pleural effusions were receding. The ascites was diminishing. He got off O2 support. And so a second round of metagenomics showed that the bugs, well, Pseudomonas in particular, was gone. And he made a pretty rapid recovery right after that. So he was home after 5 weeks in the ICU, got reunited with his fishies, which he just loved. And then a week after that, is Father's Day, actually took him down to swim with the dolphins in Florida because he just loved fish. That was Bertrand. You know, and I share this because I think a lot of us as parents have had an experience that's somewhat like this, these harrowing ICU stays. But for me, I guess I'd always known that Bertrand was medically fragile, but what I realized was that Bertrand had moved firmly into bonus time. If he wasn't there already, he was there now, that his life was at risk at any moment. And I moved as fast as I could. You know, I got a month later a clinical trial funded to do a new proposed therapeutic for fringilla 1. This was in conjunction with Mayo. I actually got him up to Mayo in February of 2020 to do the safety checks for this trial to kick it off. Then, of course, the pandemic hit. And unfortunately, over the next year, his health just continued to take hits. It was, we couldn't get out, we couldn't do anything. He kept going back to the ICU. He was suffering from adrenal insufficiency. And, it was just a long decline over the whole year. And I realized for quite some time that, I didn't know how long Bertrand had left, but I knew it wasn't unbounded. I knew that was measured probably more in low years than long years. And towards the end, I just there wasn't anything specific that told me that he could lose his life. I just felt like he was in a very precarious situation, and so I actually launched a little sunrise tour on the weekends. I took Bertrand and his siblings off to places to rest, relax, and see beautiful things. So we saw sunrise at beaches and at mountain lakes and in the mountains. And of course, we did things like pillow fights at nights, and as much as I could just to see him happy. Again, I didn't know anything was coming, but just 2 days after this picture was taken, he went to the ICU again. And in the after 48 hours, literally the shortest ICU stay of his entire life, the shortest hospital stay of his entire life, he was gone. It was sepsis. It wasn't COVID. It was. And I started the whole process. I started collecting the material. We started doing the metagenomics, and before I could even get the results back, he was gone. And so it was just a total shock. Even now, I think about how fast it was, and it's just hard to believe. But because I did that little sunrise tour, I've always tried to find the sunrises ever since. What was interesting to me though was that right after he passed, the very next day, there was no sunrise. It was permanent fog over Birmingham for almost a week. But I was driving past that morning at the park I would often walk him in. I just jumped out and started taking pictures of this sort of eerie golden glow and of the fall foliage, which was just setting in. And, really didn't see the sun for almost a week. It was really kind of astonishing. But it kind of fit my mood at the time. The sun wasn't rising and neither was my son. So it fit the moment. And I've been reflecting a lot since on what it's like to be a rare parent. And I found something I wrote back in May 2012, which I think may resonate with all of you. This is on the blog that we maintain for him. And it says that when you're a rare parent, it means fighting when you know you cannot win. It means believing the crippled will stand and walk. It means believing the mute will speak. It means believing the blind will see. It is about never yielding to the impossible when your child is on the line. It is about exerting a force of will so strong that even if only for the briefest of moments, the line between the possible and the impossible bends. It is about exerting that force for so long and with such intensity that in rare circumstances, that line breaks. And I think what's amazing about all of you in Peer is that you're here because you're that parent. You're here because you are trying to do the impossible for your child. And I think that is absolutely beautiful. And I guess, what I'll say at the end, when it comes to, what really matters is that, lead that fight as long and as hard as you can, but also focus on the moments, the happy moments you can have with your child where you don't think too much about the past or about the future, and just enjoy that time that you get with these very special children. And since I've had a lot of time to think about, what comes next, how to maintain an active legacy. So one thing I've done, for example, is created this very special perpetual endowment at UAB. And the way it works, only the interest can be used to help patients, but it has to be used in ways that, Bertram was serviced in his life. So basically it's for science and the service of patients. And it's not a huge fund yet, but it's big enough now that the interest was sufficient to start paying for an experimental medication we made for one patient, and it actually worked. So he's already made a huge difference through this for at least one patient, and I hope many, many more to come. So that's what I've got for today. And I did mention that I would give you a pointer to the algorithm. So you go to Bertrand's site. I've turned it into a hub to collect information, a bit about him, but also over time, a place where people can go for resources if they should find themselves in the same position that I was 13 years ago. So that's all I have for today. I really thank you for your attention, and just wanted to know it's a pleasure more than ever, and always to speak to your patients' parents, because I feel like I will always be one of you.
Well, I'm going to be left a little bit tongue-tied after that presentation, Matt, and I don't think anyone envies me. I think there's probably not a dry eye in the house, but it's even knowing a lot about your family story and the work that you do. It's still quite incredible to hear you put it all together. And I know I speak for all of us, that we're so grateful that you've taken the time to do that for us, and in such a personal way, because I think, this resonates for certainly for me, in so many ways, personally and professionally. And so we just have a couple of minutes left. And so I'm going to take moderator's prerogative to ask you a question that I think also aligns well with questions that have come in through the chat or through the Q&A, which is, your story and Bertrand's story, take place at this incredibly vibrant time in precision medicine. Where we were 10 years ago is so dramatically different from where we are now. And I'd love to just hear you speculate where you think we might be 10 years from now.
Yeah, well, I love to speculate. And, if I look at the forces at play and the science that's happening, there's always things that are going to happen that we can't predict, but all those will really do is add more options, I would say. But even if you just take present trends and extrapolate out, I think we are entering into the era of nucleic acid therapeutics, or you may have heard these as oligonucleotide therapeutics, or antisense oligonucleotide therapeutics, or ASOs. I think their era is here. The FDA is catching on. They sort of fit the right profile in terms of cost and complexity of development. And I think we're over the next decade, I would speculate we're going to get a handle on this to the point where it starts to become a realistic option for truly individualized medicine. Right now, it's still extreme. It's not scalable enough yet to do everybody. But if you extrapolate out, I think we're going to get there. I would also venture to guess that over that same timeline, there's enough movements afoot for gene therapy and gene editing that within 10 years, it might start to become more practical on an N-of-1 basis. But I'm still much more optimistic about things like ASOs. And I also think that we'll see an explosion in drug repurposing because it's one of the easiest things, and we're getting better and better at doing it.
Mm-hmm. Mm-hmm. Thank you for that. And just in our last minute, I would be neglectful if I didn't ask if there was anyone on the call who was interested in talking seeing if some of the tools that you're using could be applied to their case. What's their next step? Yeah, so your next step is you can email me. Best if you also tag somebody else at the Institute, because to be honest, my inbox is just overloaded right now with parents and patients asking for help, and I do eventually get around to everybody, but it can take a long time. So if you tag assist@mite.net as well, that will make sure that I get back to you, hopefully sooner rather than later. But yes, I would like to help everybody, and I try to help everybody eventually. I just apologize that I'm still working on the scalability side of this, and the Institute helps me a lot, but we don't have nearly enough resources yet to help everybody in real time, but we try to help everybody eventually. Well, I think that's a great place to leave it, as I know we all share that goal with you, and look forward to collaborating with you and helping to build that scalability in the future. And I'd just like to join everyone who's putting comments in the chat and in the Q&A. Huge thanks to you again for sharing your story and for joining us today. And with that, we'll let you go back to all of the things that you have going on. And thanks again.
All right. Thank you all so much. It's been a real pleasure.
Thank you.